So my favorite group, the Excavates. Again, these are the organisms that lack mitochondria. Typically. They fall into several groups. The Metamonads include the Diplomonads, including Giardia, which is an organism you may have heard of before, particularly if you're a camper. The Trichomonads, which I'm sure you've never heard of before, right? Oxymonads, these are the termite gut symbionts. Reclinomonas is an amazing organism. Naglaria is the brain-eating amoeba. And Euglena and Pannasoma are in the Discobians, yeah. The Excavates have a well-defined oral groove, and so they pull food, typically bacteria, into this groove. That's the excavation that they're talking about. These names are kind of weird. Like I already said, they lack mitochondria, but they clearly have something, either something that's a relic of a mitochondrion, like a mitosome or a kinetoplast, which I'll talk about in a minute, or at very least they contain clear signals in the genomic DNA that mitochondria used to be there, because most of the mitochondrial genes in most organisms have migrated through the nucleus anyway, and those genes remain in these organisms. And I mentioned that probably the root of the eukaryote tree is amongst these creatures. The Discopans are a really interesting group. These organisms really do have the most primitive mitochondria of all. There's usually only one in the cell, so consider it a symbiont. And they have complex genomes that really do look bacterial. Let's see. Heteropogonia are Naglaria's in there. And the euglenozoans include two kinds of creatures. Euglena, you guys have probably all seen euglena, right? Every grade school in the world shows the euglena. It's pretty, it's got a big flagellum, it moves sometimes in an inchworm kind of fashion, bright green, really pretty. And it's pretty scary to think that that thing is actually a pretty close relative to Pannasoma, the causative agent of sleeping sickness. These things lack mitochondria. They have instead a kinetoplast, which is a very highly derived mitochondria. It's got DNA in there that as a structural unit component. Not only does it encode genes, but they actually form part of the scaffolding and structure of the organelle. DNA isn't usually used in that way, but it is here. Gene expression in these things is really interesting. There's a bunch of RNA editing where the primary transcript is very heavily edited after transcription by the insertion and deletion, usually of U's, but also other things. Also in here is leishmania, which has been in the news a little bit recently. Leishmaniasis is a pretty awful disease. Chagas disease. Here's my favorite one, Reclinomonas americana. I collaborated with a guy looking at the mitochondria of this thing. If you want to know what this organism really looks like, the pictures don't do it justice. Imagine a couch potato laying it in a wine glass. All right? The shell looks just exactly like a wine glass with a basal plate. And the creature itself kind of lays in it. That's where it gets its name, Reclinomonas. It's got two flagella, one of which is usually in the oral groove, and the other one beats to drive things into that. Freshwater creature, and again, we already talked about the mitochondria. Most of you know something about this creature, Pannasoma. This is the causative agent of sleeping sickness. It's an obligate parasite. Complicated life cycle. It lives in the tsetse fly and in mammals. And the creature itself, it has very different structure in these two. And so there's This is what it looks like in animal blood, and it looks very different and has very different gene expression in the fly gut, which is where it lives. It also migrates to the salivary glands, of course, in the fly. That's how it injects it into you. I won't go into this creature very much. It's a pretty devastating disease. It's one of these diseases that doesn't get the interest it should because poor people are affected by it, right, and not rich people. The other thing is that these diseases are harder to deal with than a lot of others because it's a eukaryote. Things that kill eukaryotes are usually pretty toxic, right? The Metamonads are a group of really, really interesting creatures. They are structurally, on one hand, a little more complicated, but on the other hand, a little simpler than most eukaryotes. So they have this axial fiber. They've got some complex structural insides. They've got this is Giardia. It's got this smiley thing that nobody knows what it does. And it's got a ventral sucker. These sort of structures are common in this group of organisms. But in terms of the typical eukaryotic cellular structure, they're very different. They lack Golgi. They lack endoplasmic reticulum. They lack mitochondria, or have only relics of them. These are clearly derived characteristics. And in most cases are probably the result of evolutionary reductive evolution. Because they're parasites. Giardia is a causative agent of runner's diarrhea. If you go to a campground and get sick, it might be Giardia. It's pretty bad. People get really bad diarrhea and can get dehydrated. It can be a real problem. It's really interesting. The life cycle is really cool. It's binucleate, and all the organisms in this group are binucleate. So here are the two nuclei. The chromatin is condensed all the time. So that's why it's such a dark spot. The nuclei are actually quite large here. And so instead of stopping, if you think of the normal cell cycle, the nucleus divides, then the cell divides, and then you stop. And when you're ready to go again, the DNA divides, the nuclei divide, then the cell divides. These organisms go through the same cycle, but the stop point is at a different place. And so in this cell, the first thing that happens is that the cell divides, separating the two nuclei into two separate cells. Then the DNA divides, and then the nucleus divides to create a binucleate form. So the stop point is before cell division instead of after. I show this one because it's just really cool. It's an Nazimonad. It's a gut symbiont of termites. Almost nothing you see here is the actual protist. Only the flagella here at the end are the actual protist. Everything else you see are bacterial symbionts. This thing is, as far as I know, the world's expert at bacterial symbiosis. And so this is a cross-section through this thing that shows you what the creature looks like. It's just the central cell body. There are a number of specific internal bacterial symbionts too. It contains these veins that stick out, and then all these rod-shaped bacterial symbionts that go along with it. There are at least 3 different kinds of symbionts on the outside, plus the ones on the inside. That's in the central vacuole. You can't grow this thing, so as far as I know, still, it would be simple, but it would be relatively simple to mechanically pull one of these things out of some gut contents of a termite wash it off, and do a high throughput sequencing, right? And find out who all these creatures are. Go back with probes and identify them specifically. As far as I know, that's never been done. All right, I think that's it for today.